right, this is a chapter of the book, besides the nutrition chapter, that doesn't necessarily just relate to one system. And it's a wonderful place to look at this chapter because what we've just looked at in the urinary system is how the kidneys affect the water balance and electrolyte balance and so forth with your body fluids. But you've learned that blood plasma doesn't always stay in the same place, does it? Sometimes it's interstitial fluid, or it may be inside the cell, cerebrospinal fluid, filtrate. So it's constantly moving from compartments. So in this chapter, we're going to pull a lot of that together that we've talked about throughout at A&P, looking at water levels in the body. And as we look at water levels, we'll be seeing the substances dissolved in it that affect electrolyte levels and then your pH, the acid-base balance. Anything that affects electrolytes affects water and vice versa. Does that make sense? And it's because of the osmosis, basically. um, all right. So if we're going to start looking at where are your body fluids, and some of the bulk of your body fluids are actually inside your cells, and that would be your not inter, that's in between cells. Intracellular is inside your cells, and then everything else is considered extracellular fluids. But of the extracellular fluids, you have a lot of different kinds, like interstitial fluid, that's the intercellular, isn't it? Or tissue fluid in between your cells. Most of your extracellular fluids are in that compartment, interstitial fluid. You need to make sure you know that. Okay? But then you also have blood plasma is another extracellular fluid, as is lymph. And then all these other ones that are lumped together as transcellular because they're bound by special compartments or membranes. Okay? It's like cerebrospinal fluid, the fluids in your eyes, vitreous and aqueous humors, the synovial fluid in your joints, and then the serous fluids between visceral and parietal membranes. Okay? There's a lot of graphics, and I haven't put them all in your notes, but you'll see a lot of tables and graphs in different ways in your book that shows just in a picture the percentages of these different ones. You don't have to know exactly 63% and 37%, but you need to know that you've got more intracellularly than you do extracellularly. And of the extracellular, the most abundant would be interstitial fluid. Okay? And know which ones are listed as transcellular. So what I have done in your notes is made a very brief explanation of what's in this chapter. Okay? But if I wrote it in your notes, you better know it. You know, you can see the notes, the bulk of it is just a bunch of pictures, but they explain some of the things that I typed. Okay. All right, so when we look at your extracellular compared to your intracellular fluids, they have different concentrations of certain ions, like your extracellular fluids. You can see I've listed some ions: sodium, chloride, calcium, bicarbonate ions. You have more of those in extracellular fluids than you do intracellular. And with the intracellular, you have more potassium, magnesium, phosphate, PO4 is phosphate, and then SO4 is sulfate ions. But do you notice for each of these, I bold printed just one? Do you see that? Those are the ones you need to know for the test, what I just circled. And that should be super easy for you if you remember about way back in AMP1, a resting neuron. Remember when a neuron is at rest? What ion was highly concentrated outside the cell? Sodium. What ion was highly concentrated inside the cell? Potassium. See, intracellular has a high potassium level. Extracellular has high sodium. That's the easy way to remember it. Go back to think about neurons. Okay. All right. But then you have some other ones too that are more abundant in one place than the other. But for your test, know the sodium and potassium. Okay. And then blood plasma is unique compared to all your other body fluids because it has more proteins. Okay. And we've learned that. Proteins stay there because they're too big to cross, right? Okay. Then this graph is interesting. They're showing a lot of these ions that are listed in the notes. But if we look at just sodium and potassium, I mean, this is very eye opening. The blue bar represents extracellular. So see how the sodium level is so much higher outside the cell than inside? And the potassium is so much higher inside than outside. Do you see that? Um, that has to do with that sodium potassium pump. It puts it in those concentrations. Here's another way to depict all of the body fluids. Intracellular is higher than all the others, and then of all the others, interstitial is the most abundant. So that's just another way to show it. Okay. All right. And if you haven't learned anything, I hope you have learned the difference in hydrostatic and osmotic pressures. Those two pressures are the driving pressures for water movement in the body, as water moves from one of these compartments to another, right? Which one is a pushing pressure for fluid movement? Hydrostatic, that's the pushing. So the higher the hydrostatic pressure, the more water moves away from that area of high hydrostatic pressure. Osmotic pressure tends to keep. That's a keeping pressure. Or you can say pulling. I'm not going to write that down because your authors of your book might be horrified to say that that's a pulling pressure. But water is drawn to an area of higher osmotic pressure, and those pressures oppose each other, don't they? If they're in the same compartment, right? And so you have to see what overrides the other. But that's how substances are moving, either because of the pushing action of hydrostatic or the pulling action of the osmotic. That's how the water moves. And 9 times out of 10, that water has something dissolved in it, doesn't it? So if we look at water balance in the body, this might surprise you that at any given moment of today compared to any given moment of tomorrow, you have about the same amount of water in you always. Okay? What you drink, what you take in that day is going to be equal to what you eliminate, okay, on a daily basis. So because of that, because what you take in is equal to what you output, your water amounts in you stay pretty constant. Okay, and so how do you get water into your body? You know, water is the most abundant chemical in you. As a third of your weight is water on the average. But most of what water comes into you is in the form of what you drink. That's obvious, isn't it? But you also get some water from foods that you eat. So I've starred this, meaning that's the most way. Okay. All right, but you also get it in certain foods. You know, some fruits have a lot of water content, right? You also get water from some chemical reactions. Like, do you remember dehydration synthesis? Like you take 2 amino acids, join them together. In order to do that, you have to pull out water. All right, you've just made a water molecule. So you get some from chemical reactions in you. All right. The main mechanism that controls our intake of water is your thirst mechanism. I've described that earlier when we were looking at ADH and the hypothalamus. Hypothalamus doesn't just signal ADH release, but it also controls your salivary glands and making you thirsty, so you'll take in water. Okay. So this is just another chart. It's showing that the bulk of what you get when you get new water in you is from what you drink. Okay. You don't have to know the exact percentages. Just know the main one is through drinking. Okay. What about your output of water? The main one is through urination. Okay. If you have too much water in you, you're going to urinate it. We just saw that, didn't we? Okay. But you also lose some water through your skin and through lungs. Every time you breathe out, you're breathing out some water vapor, aren't you? And then through your feces, you lose a certain amount of water. And through sweating. Don't worry about sensible and insensible perspiration, the difference. Sweating is one, just normal. Sometimes you just lose some water through your skin, not through the sweating action. Right, that, and then through exhalation, you lose it. That's insensible perspiration. All right, and then one of the main controls that regulates how much water you eliminate through your urination is ADH, and we just talked about that in the last chapter. Okay. All right. So we've looked at that. See how this is pulling together a lot that you already know, but it's making it all make sense. Okay. All right, and this is just a graph showing you that you lose most of the water every day through urination. So again, you don't have to worry about the individual percentages. Okay. All right, and there are a lot of water balance disorders. I'm not going to test you on these, but they are interesting, and I thought I put them in here so that maybe you'll go into your book and read about those, because you hear about people struggling with dehydration. You know, whether it's an athlete or you're working outside for long length of time and not getting enough water, or you know, somebody maybe has diarrhea. So in a medical setting, people can dehydrate for various reasons. You may have even heard of water intoxication, where somebody takes in too much water. Well, that messes up the water levels. It makes it more hypotonic than it's supposed to, and so that affects that movement of water. And so your book talks about that some. And then edema. We've talked about edema in the past, and that's a fluid issue in the body. It affects your water levels. So, but again, I'm not going to test you on these, okay? All right, so we've done water. Now we're going to look at electrolytes. Where do you get most of them? You take them in mostly from the foods that you eat. You get some electrolytes from beverages. You know, soft drinks has some electrolytes. If you drink Gatorade, it's got a lot of electrolytes, but mostly it's from the foods that you eat. Electrolytes, remember, are just all these different ions. They're in a compound form in the foods that you eat. And then you get rid of excess electrolytes through urination. That's the main way, so I start it. Make sure you know that's the main way. But also you get rid of some through sweating and through your feces. And then there are a lot of regulations in the endocrine system that affect electrolyte levels. But I'm only putting two here for your test. And those are aldosterone, that affects sodium and potassium levels. Okay, if you increase your aldosterone release, then your kidneys are going to reabsorb more sodium. And remember, sodium and potassium always move in opposite directions. So if you reabsorb sodium, you secrete potassium at the kidneys. So a stimulus to your adrenal gland to cause you to make aldosterone is an elevated blood potassium level. Because if it's elevated, that's an indication the sodium is low. Okay, so you want to save all your sodium and secrete the potassium. And then PTH, remember that is parathyroid hormone. And it causes your blood calcium to rise through a lot of actions. One of those, it activates osteoclast. But you have another one that affects it, and that would be calcitonin, causes calcium levels to lower. But this is the one on this test. Okay. All right. So you have a good electrolyte balance when the electrolytes you take in is equal to those that you eliminate every day. And so this is just another way to show you electrolyte intake. Most of that is from foods. Electrolyte output, most of that is from urination. Okay. If you take in more of a certain electrolyte than you eliminate, then you have an imbalance. And a lot of medical issues result from electrolyte imbalances. And those, when you get into your future programs, like nursing, you will spend a lot of time, class time, talking about electrolyte imbalances, because they're going to affect fluid movements. They also affect what that electrolyte does, what its significance is. So here, we're not going to dwell on those. Your book may talk about them some, but you just need to know where you get electrolytes and how you eliminate them. I'm making this really easy for you. Okay. But this chapter, especially if you're in nursing, this chapter is a big reason that you don't need to get rid of this book, because it does explain some of this topic, fluid fluid levels, electrolyte levels. It explains it probably in a way that's easier to understand than the more technical ones that you're going to get into later on. So it would be good to have this chapter available to you. Okay. So here, this is just showing you a cascade that occurs when your electrolytes are out of balance. Okay, for example, on this left one, this is just showing you what I've already mentioned, that when your potassium levels are high in your blood, that signals the adrenal cortex to produce aldosterone, which then goes to your kidneys and causes you to release the potassium through tubular secretion, and then in turn to reabsorb sodium. And then on the on the right, it's showing you if your blood calcium ion levels decrease, that's going to signal the parathyroid glands to produce parathyroid hormone, which has a lot of target cells, but all of that helps to raise your blood calcium. Okay. All right, and so we're going to finish up. We've looked at water levels, we've looked at electrolyte levels. Now we're going to look at your acid-base balance. It is extremely important that our pH in our body fluids remains where it needs to be, because if the pH is altered too much, enzymes will not work, right? Remember, enzymes are specific for certain pHs. And so we have chemicals in us that can raise the pH of a body fluid or can lower the pH of a body fluid. Those that lower the pH are called acids because they release hydrogen ions, and the more hydrogen ions, the lower the pH number, right? The more acidic. Remember, pH ranges from 0 to 7 to 14, and the number, it's opposite. The number is an opposite reflection of hydrogen ion levels. The lower the number, the more hydrogen ions, and the more acidic it is. Y'all remember that? It's a logarithmic scale, but more hydrogens, the lower the number, the more acidic, and less hydrogens, so less hydrogens, or more of another hydroxyl ions, the more basic it is, or alkaline, basic or alkaline it is. All right, anyway, the chemicals that release hydrogens are called acids. The chemicals that release, the substances that release chemicals that combine with hydrogens are called bases. Back in A&P 1, I taught, for those who had me in A&P 1, that bases were substances that released hydroxyl, OH negative, that's a hydroxyl ion. But that's a very narrow definition of a base. A much more generic definition is a base releases a substance that combines with hydrogen, which is going to raise the pH because it's lessening the hydrogen ion concentration. Well, don't you see that that would be attracted to a hydrogen ion to form a water molecule? So do you see how it takes up hydrogen? Okay. And so it makes it become more alkaline, right? Because it's lessening the amount of hydrogen. Okay. All right, and we have acids and bases in us just automatically. So, but some of our acids that we have in us are strong acids, some are weak acids. Some bases are strong bases. Some bases are weak bases. So to give you an idea of the difference in the two, let's say if here's a beaker of water, okay, and water is going to break up into hydrogen and hydroxyl. Some of it will. Not all of it does. A lot of it stays intact, but a little bit will break up. At this point, we have equal hydrogen and hydroxyl, and that would be a pH of 7.0 if you have equal hydrogen and hydroxyl. Okay. If you had a strong acid, like hydrochloric acid is a strong acid, so let's just pretend I have 12 of those. Because it's a strong acid, what that means is it will release a lot of hydrogen ion, not just a little bit, but a lot of it. So let's say 10 of these break apart. So do you see how that would release 10 individual hydrogens? And then you'll have 2 that stay connected. You also have 10 chlorides. But anyway, do you see how that changes the pH a lot? That would be a strong acid. And a weak acid would be one that still ionizes or dissociates in water. But let's say it's like carbonic acid is a weak acid. So if 12 of those dissolve in, maybe only 6 of them break up, or 4 of them break up. If 4 break up, you're going to have 4 hydrogens, and then 4 bicarbonates, and then, well, there'd be 8 of them remain intact. So it still lowers the pH because you're adding more hydrogens, but it doesn't lower it as much, right? Okay. That would be a weak acid. Okay. Strong bases and weak bases are the same way. One of them, a strong base, is one that takes up more hydrogens than a weak base does. So which one affects the pH most quickly, a strong base or a weak base? The strong one. Okay. If I told you I was going to inject an acid into your blood and you didn't have any control over that, I am going to inject an acid into you, but you could choose which acid it was, would you choose a strong one or a weak one? A weak. It wouldn't change the pH as much. Changing pH is bad for you. Okay. It wouldn't change it as much. Okay. All right. But we have mechanisms, even though we produce acids all the time and we produce bases all the time, we have mechanisms that help prevent drastic changes of pH so that our pH levels stay like they need to. All right. You might be surprised at how many acids you produce on a daily basis. This is just some sources of hydrogen ions that can drastically affect your pH. Like every time you're breathing, whenever you undergo cellular respiration, you produce CO2, don't you? And when CO2 gets in your blood, it combines with hydrogen to form carbonic acid. It's a weak acid, but it's still an acid. It's going to change the pH, isn't it? Okay. When you have anaerobic respiration of glucose, you produce lactic acid, which is an acid. It changes the pH. It releases hydrogen. When you break down fatty acids, a result of that is something called a ketone body. It's an acid. It releases hydrogens. When you break down proteins that have amino acids with sulfur in their R groups, that sulfur can combine with hydrogens and oxygens to form sulfuric acid, which is a strong acid. So that's a change in pH. And when you break down proteins that have phosphorus in their R groups, in some of their amino acids, and also when you break down nucleic acids, they have phosphorus. That forms phosphoric acid, which can change the pH. Isn't that shocking? How many acids we're making all the time? But thank goodness we have mechanisms that help prevent a drastic change in pH. Okay, and that's what we're going to be looking at towards the end of this chapter. And these are some buffers. You hear about buffers. If you were in a chemistry lab, how many of you have taken a chemistry class and you've been in a lab and you've used a buffer? Okay, what was the purpose of that buffer? Do you remember? It was to regulate the pH, wasn't it? To keep a pH constant. And you have buffers in you that automatically prevent drastic changes in pH. And some of them are chemical buffers, and some of them are physiological buffers. All right, but the combination of those help prevent you from experiencing what we call acidosis or alkalosis. Those words you might remember way back from the chemistry chapter 2 in your book, when we were first talking about pH. Okay, most of your body fluids, pH is slightly alkaline. Like your blood pH needs to be right around 7.4. Okay, and so that's your normal pH range. Pretty much everywhere except the stomach, and its pH is 2 because of pepsin has to work at that low pH. But everywhere else, it's got to be a little bit slightly alkaline. You can survive, not super well, but you can survive in that pH range, 6.8 to 8. Okay. But the desired is that right around 7.4. Okay. But because we're constantly bombarding our fluids with acids and bases, that can change. Okay. And so we have these buffers that prevent drastic changes. Um, so here, if you increase your concentration of acids or decrease your concentration of acids, do you see how it can cause pH to drop or pH to rise? That's just showing you that. You won't have to explain all of that, okay? Um, all right, and then now you hear this term. It is not on your test. I probably should put it on this test. I'm not doing justice to this chapter. But you hear the word acidosis and you hear the word alkalosis in a medical setting, okay? And you can have respiratory acidosis. Your blood pH can drop. It has to do with if you've got some respiratory issues, like somebody may have COPD. That's a respiratory issue, which they're not breathing out enough CO2, and if they don't, that accumulates CO2 in their blood, and an excess of CO2. Remember how CO2 combines with water to form carbonic acid that splits up into hydrogen and bicarbonate. You see how that's going to raise your hydrogen levels? Okay, that's respiratory acidosis. There's metabolic acidosis. You hear about that. It could be from kidney failure. Diabetes can trigger it. There's metabolic, and then there's respiratory. Again, I'm not going to test you on the difference in those, but you hear about that in a medical setting. These flow charts are wonderful for you later on in other courses, and I'm sure they'll be in those books too. But this is just a real simplified version. It's good to get the first time. When people have diarrhea for a long period of time, they're losing not only a lot of water, but they're losing a lot of normal bases, which can trigger metabolic acidosis. See? Or vomiting, same way. So you can have alkalosis, respiratory alkalosis, metabolic alkalosis. That's when the pH rises. And here's some examples of what can cause all of that. We've talked about hyperventilation in the respiratory system. Hyperventilation can cause you to lose too much CO2. Remember, you're breathing out more than you take in. Okay, and so that's going to drop your hydrogen ion level. Okay, and then the metabolic, you can see. Metabolic alkalosis can also deal with digestive tract issues. So how do we prevent these changes? That's this last part of the chapter. We're going to look at these buffer systems. You've got a chemical buffer. That's the one that works very quickly. Doesn't have as long-term of effect as the second one, but it's really good. And this is the first way your body prevents a big change in pH. You have chemicals in your body fluids that are described as buffers. And basically what a buffer does is that it combines, like if it's an acid, if you've got acidosis going on, it can not prevent the change in pH that the acid causes, but it makes it less drastic, less of a change. Because it basically combines with strong acids to cause them to become weak acids, so they don't ionize as much. Okay, or they could combine with a strong base to make them a weak base. They don't combine with an acid and change it into a base, though, or vice versa. So they make a strong acid a weak acid, or a strong base a weak base. So do these chemical buffers prevent a change of pH? The answer is no. They just make it less drastic of a change. There's 3 kinds of chemical buffer systems. I'm not going to go test you on the chemistry of the 3 systems, but you do need to list them. There's the bicarbonate buffer system, the phosphate system, and the protein system. I'll look at it with you a little bit. Here they're laid out. As far as this slide is concerned, you just need to know their names. These are chemical buffer systems. These are chemical buffers. Just like it said on the other slide. Okay, chemical buffer systems. Okay, the bicarbonate buffer system obviously involves the bicarbonate ion, and the two chemicals of the bicarbonate buffer system are the bicarbonate ion and then carbonic acid. So let's say your bicarbonate ion, it's one of these bases that very quickly combines with hydrogen. So let's say you've had an acid, sulfuric acid is in you from the breakdown of some of your proteins. You're releasing a whole bunch of hydrogen ions. Well, the normal bicarbonate ions that are in your body fluids are going to combine with that. And so it takes the effect of, say, sulfuric acid, which is a strong acid, and changes it to the effect that carbonic acid is going to have. See, it makes hydrogen, but remember, carbonic acid is a weak acid. Okay. At the same time, if you had a strong base in you, like you have excess of these, they're going to combine with hydrogen to form the carbonic acid, which is not going to release it as much. It's weaker. So it goes back and forth and back and forth. So basically, it's converting strong to weak. Okay. The phosphate buffer system works exactly the same way as the bicarbonate buffer system. It just uses different chemicals. Like there's dihydrogen phosphate and monohydrogen phosphate. Those are the two chemicals, which you don't have to name them. Just know phosphate buffer systems. But one takes the effect of a strong acid and makes it as a weak. The other takes the effect of a strong base to make it weak, okay, preventing a drastic change in pH. And then the protein buffer system, I love this one. It's neat. Every protein in you can act as a chemical buffer because, remember, proteins are made up of long chains of amino acids. And do you recall that on one end of an amino acid, you have an amine group? And then that's the NH2. And the other end is a carboxyl group. That's that C double bonded to an O and then to the OH. And so that's one amino acid. So if you have one amino acid connected to another one, to another one, to another one. So let's say this is a protein I'm making. On one end of the protein, you have a free carboxyl group. On the other end, you've got the free amine group that's not bound to anything else. All right, what's naturally going to happen is that this hydrogen breaks off and comes over and attaches right there, the ion of it. Attaches right there. And so do you see how one end of that whole protein becomes positive, the other end becomes negative? Do you see that? So now, let's say that's a hemoglobin. Hemoglobin can act like this. So let's say if there's excess hydrogen in the body fluid where hemoglobin is, like in your cell, do you see how this would be attracted to the hydrogen? So that will lessen the hydrogen amount. Or if the pH rises too much and a body fluid needs more hydrogen, that can be released right there. So just a protein itself can act as a chemical buffer. That's pretty neat. But you just need to list, there's a bicarbonate buffer system, protein buffer system, phosphate buffer system, and their function is to convert what to what? Strong acid to weak acid and strong base to weak base. So they prevent a big change in pH. They don't prevent a change. They just make it less drastic. Okay? All right, so that's your first line of defense. First time you have a change in your pH, these chemical buffers kick in. Your second line of defense, which is more long-term for you, it may take a few hours or days for them to actually kick in, but they last a long time. But that's your physiological buffers. We have talked about these throughout A&P. One has to do with your breathing, your respiratory center. You know, your respiratory center is greatly influenced by hydrogen levels, isn't it? Okay. And then the other is your renal mechanism, which we talked about in the last chapter. At your kidneys, Your last effort to get rid of excess hydrogen is that tubular secretion. You can secrete any excess hydrogen from your blood. Okay, so just be able to list those. And here it's shown just in a little chart. You got your 3 chemical buffers, your 2 physiological buffers. The chemicals are the first line of defense, which act immediately, but they're not as long term. Second line of defense takes longer to kick in, but it's more long term. Okay? So I apologize. I know you're probably happy. I apologize, though, that I haven't gone into as much depth with this acidosis and alkalosis, but for this unit, I want you to really dwell on the kidney and what it does.